A-Level Biology 9700, Paper 4, Variant 2, February March 2026. And this is the second video on this paper in which we are going to discuss question number 3 and 4. And question number 3 is Huntington's disease is caused by a mutation in a gene coding for protein that has a number of cellular functions. So identify the gene and state the name of the protein that are affected by the mutation that causes Huntington's disease. So the gene name will be HTT. So that is from Huntington's disease. So HTT is for this, and this protein is HUNTING Huntington. So this is the name of that protein. Now the next question is, in many countries, a genetic screening test for Huntington's disease is available. Adults over the age of 18 years who are considered to be at risk of developing the disease are offered the genetic screening test. So suggest which adults are considered to be at risk of developing this disease. So here I would like to say that uh, actually the individuals which are which have a history with this disease, they can be the risk uh, on it. So as for example, I would like to say who adults who have a family history of this disease, they having a parent, sibling, or close relative with the condition or known well allele, so they will be at high risk. So I would like to write this answer as next question is suggest and explain why the genetic screening test for this disease is only offered to adults who are at least 18 years old. So individuals who are must be legally adults to give informed consent and make an autonomous, fully understood personal decision regarding predictive testing. So this disease typically has an adult onset. As for example, I would like to say that the symptoms usually do not appear until age 30 to 50 and currently has no cure, so testing a minor offers no immediate clinical benefit while posing significant psychological emotional distress. So this is the reason that they do with the people who are at least 18 years old. Individuals legally must be adults who can give informed consent and are able to understand that what they are going to do. Secondly, the symptoms of this Huntington disease appear after 30 to 50 years. So, testing a minor offers no immediate clinical benefit. The next question is part C. Part of the gene associated with Huntington's disease has a repeated DNA triplet nucleotide sequence of CAG. There are 35 or fewer CAG repeats in the normal alleles, with 40 or more CAG repeats are expected to result in the development of Huntington's disease. So the genetic screening test for Huntington's disease includes a polymerase chain reactions, PCR. This results in amplification of the gene that is associated with Huntington's disease. So the DNA fragments produced by PCR are then observed using agarose gel electrophoresis. So the genetic screening test results of 18 years old adults A, B, C, and D who had no symptoms of Huntington's disease are shown in the figure 3.1. Control DNA controlling the fragments from amplification of alleles with 35 and 40 CAG repeats is also shown. You can see they have the adults A, they have moved far. Adult B, little bit behind the A, and C is almost very behind, and D, you can see them. So in this way, the controlled DNA is also there. So the results of the tests were used to make predictions. So adult A will not develop Huntington's disease. Adult B may or may not have developed Huntington's disease. C and D have developed this disease. Adult D will develop this disease before adult C. So explain how the results of gel electrophoresis shown in figure 3.1 support the predictions. So here I would like to go with the figure 3.1. You can see in figure 3.1, before going this detail, I would like to say that the shorter DNA fragments uh, can migrate further faster down the gel towards the positive electrode, while longer DNA fragments, more CAG repeats, migrate less far, stay closer to the top. So this is the actual explanation. So what happened over here, I would like to say that the adult A, you can see it has moved. It has moved further than the 35 repeats. So you can say uh, uh, this control band is there. Control band is there, the last one. So it is moved far and faster than the control band and other as well. So you can see the B may or may not develop, has one band located above the this A. So there is the ambiguity about this, or you can say the doubts that whether they will be produced or not produced. And what about the C and D? You can see the C and D left behind the, so it means they have more repetitions. C has less than D. So they have repetitions more, so they have larger fragments. So in this way, they have not moved farther away. So they have significantly more CAG repeats than adult C. So the athlete distance stayed highest in the gel. So I would like to say the earlier onset for adult D, a higher number of CAG repeats correlate with an earlier age of onset and greater disease severity. So it is the result which I would like to go over there. So I'm going to write this result on the given space. So as shorter DNA fragments migrate faster down the gel towards the positive towards the positive electrode. While longer DNA fragments migrate less far, stay closer to the top. So adult A both DNA moved faster than 35 repeat control band. So disease causing allele is not present. Is not present. Adult B may or may not develop, so has one band located. One band located between 35 240 CAG repeats. So the symptoms may or may not show. Then adult C has an allele with more than allele with 40 CAG repeats. So, and adult B has an allele with significantly more CAG repeats. Repeats. So there can be earlier onset of and greater disease diversity. Earlier onset for adult D and greater disease severity. So the next part of this question, part D, is there. Some genetic diseases such as where combined immunodeficiency SCID can be treated by gene therapy. Explained why Huntington's disease cannot be treated using the type of gene therapy used in street SCID. So here I would like to say that is the gene therapy comparison for this. As you know, the SCID SCID is caused by a recessive allele. So adding a normal functional allele restores function. Huntington disease is caused by a dominant allele, so gain of function mutation. So adding a normal allele will not stop the expression of the toxic mutant intent protein. So the mutant gene must be silenced, knocked out, rather than just added. And the third can be the SCID affects bone marrow stem cells, which can be extracted and treated ex vivo, whereas the Huntington affects whereas the Huntington's affect brain tissue, neurons, which are non-dividing cells and difficult invasions to liver viral vectors to I would like to say in vivo delivery cross the blood-brain barrier. So these are the three main explanation that why Huntington disease cannot be treated. So the first is that SCID is caused by a recessive allele so adding a normal functional allele restore function. Whereas Huntington's disease is caused by a dominant allele. Dominant allele. Second, I would like to say that the adding a normal allele will not stop the expression of the toxic mutant Huntington protein, whereas and the mutant gene can be silenced. SCID affects bone marrow, stem cells, which can be extracted and treated. Whereas Huntington's disease affects brain cells, which are non-dividing, cannot divide and difficult to control. So it was all about the question number 3. Now moving towards the question number 4. And question number 4 is outline the theory of evolution. So evolution, you know, students, this is the uh, changes which takes place with the passage of time. So here I would like to say that according to the theory of evolution, organisms within a population display genetic variations produced by random mutations and meiotic recombinations. And secondly, the environmental selection pressure act on these variations. Individuals possessing uh, advantageous alleles phenotypes are more likely to survive and reproduce. And then advantageous alleles are passed on to the offspring, resulting in a change in allele frequencies over many generations. So this is the whole process of this evolution which takes place. You can say the changes or variations which take place with the passage of time. It takes time. So I would like to write over here, that is the organisms within a population display genetic variations produced by random mutations. And meiotic combinations. Then environmental selection pressure act on these variations individuals possessing advantageous alleles are more likely to survive. Advantageous alleles are passed on to offsprings, resulting in a change in allele frequencies. Over many generations. Okay. The next part of this question is part B is approximately 95% of bird species living together today belong to an evolutionary group known as Neoaves. So, bird species in the Neoaves group are believed to have evolved from a common ancestor that existed around 67 million years ago. The neoavians that exist today can be divided into four groups. Mirandonites, referred to as Group 1, and Columbiomorphi, referred to as Group 2, and Otidiomorphi, referred to as Group 3. All other noavians referred to as Group 4. In 2014, scientists used DNA sequence data for 48 bird species to produce an evolutionary evolutionary phenotypic tree for the No, yes. So this is shown in figure 4.1. The evolutionary tree shows possible evolutionary relationships between the four groups of species within the Norovirus. So this is the 67 million years ago, not to scale present day. So they have four groups in 4.1, but the same has been given. But you can see that in 2024, after 10 years, scientists used DNA sequence data from 360, it was for 48 birds, now it was for 363 birds. So this is shown in figure 4.2. Now the question has been given on the next page, that is, describe the similarities and differences between the evolutionary relationship shown in figure 4.1 and 4.2. Move back to this figure 4.1 and 4.2. Here, first of all, I will describe and then I will write on the given space. So here you can see um, you can see the similarity in both these figures. All four groups, 1, 2, 3, 4, share a common ancestor, Norris. So they share these. And then you can see that both trees, group 1, this is the group 1. So it is divergent earlier, first from the common ancestor, and group 4 forms its own separate, and it is 4 forms its own separate evolutionary line. So these are the similarities. So if we go with the differences, so you can show a three-way divergence polytope directly from the main ancestral line, whereas figure 4.2 shows group 2 and group 3 sharing a more recent common ancestor before diverging from each other. So 4.2 indicate a closer evolutionary relationship between group 2 and group 3. This group 2 and group 3, then figure 4.1. So now I'm going to write this answer. So first of all, I would go with the similarities. In both figures, all four groups share a common ancestor. In both trees, the group 1 diverged earlier from common ancestor. And group 4 has its own separate evolutionary line. And now the differences. The figure 4.1 shows group 2 and group 3 So it is 4.2 actually. Group 2 and group 3 sharing a more recent common ancestor, whereas figure 4.1 shows group 2 shows a three-way divergence. So the next part of this question is explain DNA sequence that can be used to produce evolutionary trees such as those shown in figure 4.1 and figure 4.2. So I would want to say that DNA uh, sequences or species genes from different species are aligned and compared to determine the degree of similarities or count the number of base differences mutations. So species with fewer differences in their DNA sequence have a more recent common ancestor, so which allow algorithms to construct the balancing pattern of the tree. So I would like to write this, that is the DNA sequences sequences from different species are aligned and compared to determine the degree of similarities or count the number of base difference. Secondly, species with fewer differences in their DNA sequence share a more recent common ancestor. Which allows to construct the branching pattern of the tree. moving towards the next part of this question. State why figure 4.2 is expected to be a more accurate representation of the evolutionary relationships of birds than figure 4.1. Uh, I had told you previously when I have started this question that in figure 4.1, there were only 48 species for comparison, whereas in 4.2 is based on much larger sample size, that is 363 species. So reducing sampling error and providing more statistical reliability. So I would like to write with figure 4.2 is based on a much larger sample size, that is 363 species, compared to figure 4.1, only 48 species. So large sample size reduce error, sampling error, and provide more statistical reliability. So the next question is, part C of this question is, during the evolution of some neo-avian species, crossing over during meiosis did not occur in a large section of one chromosome for several million years. State and explain the error effect on genetic diversity of an absence of crossing over. So we have to describe the effect on decreases or reducing genetic diversity. So first of all, I would like to explain that the crossing over which takes place during prophase 1 of meiosis involves non-sister chromatids exchanging genetic material to break linkage groups and form recombinant chromosomes. So without crossing over, alleles on the same chromosome remain strictly linked. So alleles are inherited together and fixed parental combinations. So consequently, fewer unique combinations of allele are produced in the gametes. So I would go with this answer, that is crossing over involves non-sister chromatids exchanging genetic materials to break linkage groups and form recombinant chromosomes. Without crossing over alleles on the same chromosome remain strictly linked and inherit and are inherited together. Consequently, pure unique combinations of alleles are produced in the gametes. Well, student, it was all about question number 3 and question number 4. So please like, share, and subscribe my channel to watch more videos on this paper and on other papers which are coming. So thanks for watching.